Hello everyone, my name is Ahmed El-Sohemy from the University of Toronto. Today what I'd like to share with you is some of the research that we have been doing in the field of nutrigenomics, which is the science that helps us understand why some people respond differently to the same foods, beverages, and supplements that they consume based on their genetics, and how we've been able to make use of that information to develop personalized nutritional recommendations. First, by way of disclosure, I am the founder and I do hold shares in a company called Nutrigenomics, which is a U of T startup company that does genetic testing in this field. So first of all, the term nutrigenomics really is a branch of nutritional sciences that makes use of genomic information and genetic technologies to help us address those questions that we've always been interested in nutrition, and that is, how can we use it to improve human health as well as performance? Now, when we think of the experimental approaches that we take in this field, we can think of really two sides of a coin. On the one hand, we know that diet and constituents in our diet can upregulate or downregulate the expression of all kinds of genes. Diet can also cause epigenetic modifications to genes, and those are changes not to the actual sequence of the genes, but modifications to those sequences, and those can actually be inherited. Now, the flip side is what's illustrated by the bottom arrow, and what's really the focus of our research program, and that's how we can understand differences in the sequence of our genes and how that affects the way we respond to the foods that we ingest. Now, before a gene can even act on the diet that's ingested or consumed, genes can also influence the foods that we select. And so some of the work that we've done in this area looked at how genes affect various ingestive behaviors. In one particular area, why some people tend to consume more sugar than others. So this is a study that we published a number of years ago in Physiological Genomics, where we identified a variation in a glucose transporter that's expressed in regions of the brain that regulate appetite. And we found that some people who have an impaired glucose sensing ability, based on their genetics, they tend to consume more sugars on a daily basis. So Karen Enney, who is one of my former PhD students who did this work, expanded her project and looked at other genes that can also influence sugar consumption. And another gene we found was one that codes for a sweet taste receptor. So some people tend to overconsume sugar based on impaired glucose sensing in the brain. Others tend to overconsume sugar based on just their like for the sweet taste sensation on the surface of the tongue. So it's important for us to understand why some people have certain likes and dislikes to certain foods and try to understand some of those genetic drivers for some of those behaviors. Now, when we look at how genetics modifies the effect of diet and nutrition on a variety of health conditions and performance outcomes, if we trace the path from virtually any nutritional factor to any health outcome, if there have been enough observational studies, we tend to see this heterogeneity of response. So we've all heard one day fiber lowers the risk of colon cancer, another day you hear about a study that it has no effect, and yet another where it might actually increase the risk. Well, that's true for virtually every nutritional factor. If there have been enough observational studies, we tend to see this mixture of results or inconsistencies in the findings. Now, there are a lot of reasons for those inconsistencies, but an important factor is the genetics of the population that's being studied. So I've tried to illustrate here in a black box 3 potential genotypes that can help us predict if that nutritional factor will improve the health outcome, where it may have no effect, and more importantly, where the advice that we give may in fact cause harm. So clearly the concept of one size fits all just no longer applies. And we used to call these individuals that respond in the opposite manner outliers, but more and more we're seeing that these so-called outliers are much more common in the population. And so what if you are an outlier, and would you want to know, and is there something that you can do that can basically provide you with better advice than the generic one-size-fits-all advice. Now, when it comes to genetic testing, I think it's important to make a distinction between so-called disease risk genes and what I like to refer to as modifier genes. So when it comes to genes that predict disease outcome directly, those types of genetic association studies have been quite good for rare diseases. But when it comes to the common forms of chronic disease, the findings have really just been inconsistent, much like the link between nutrition and common forms of disease. And the reason is that different genetic variants act differently based on the environment. And nutrition, of course, is a very important component of our environment. Where the opportunities are really in the modifier genes. So these are genetic variations that alone do not predict risk of disease, but they affect the way we respond to various factors of our environment. An obvious example, of course, is a drug. We know that many drugs are very effective in some individuals, they're ineffective in others, and yet in some individuals we see adverse drug reactions. So the same concept really applies to nutrition. And we can use those principles of pharmacogenetics, where we try to understand how differences in the rates of absorption, distribution, metabolism, cellular uptake, and the breakdown and elimination of just about everything we ingest, much like we do in the field of pharmacogenetics. So we first started looking at some of this work by examining the link between coffee and cardiovascular disease. And at the time, there were dozens of studies that have looked at this association. Not surprisingly, results all over the map. Our interest, of course, was trying to identify genetic markers that can help us predict if coffee increases the risk of cardiovascular disease, has no effect, or where it might actually be protective. And there have been some of these studies that show this U-shaped or a J-shaped association, suggesting that moderate consumption might be protective, and as you increase consumption, risk goes up. Now, when we look at a cup of coffee, it's actually a very complex beverage, all kinds of bioactives. Some of the polyphenols that have antioxidant properties might actually be good for you. Other things like the diterpenoids that are known to raise LDL cholesterol, not so good. So a mixture of some good things and not so good things. We asked the question specifically, is caffeine a component in coffee that might trigger a heart attack? Now, when we look at how caffeine is broken down in the body, it's broken down almost exclusively by the CYP1A2 enzyme, which is one of the drug metabolizing enzymes. And it converts caffeine into this more water-soluble compound called paraxanthine, and then that's rapidly broken down into other metabolites. So CYP1A2 catalyzes the rate-limiting detoxification of caffeine. And the nice thing about the gene that codes for this enzyme is it has a common variation at this position that has a profound effect on enzyme activity, or inducibility as shown here on the y-axis. So if you are homozygous for the A allele, in other words, you inherited an A version of this gene from both your mother and your father, you essentially have a 4-fold higher rate of breaking down caffeine compared to someone who has even a single copy of the C allele. So we reasoned that if caffeine is a component in coffee that might trigger a heart attack, we would expect the slow metabolizers to be at a higher risk. So we conducted the study. It was a case control analysis in collaboration with Hanya Campos at Harvard, where we looked at just over 2,000 cases of an acute myocardial infarction, heart attack, equal number of controls, assessed coffee intake by questionnaire, and looked at their DNA from blood samples. Now, before taking genetics into account, we found that those who drank 4 or more cups a day had a 36% increased risk of a heart attack. This is the multivariate adjusted odds ratio, which means we took into account smoking, physical activity, saturated fat intake, a whole host of confounders. The real question was, is this increased risk due to caffeine? And if it is due to caffeine, we would expect the slow metabolizers to be at a higher risk. So when we separated our population, that's exactly what we found. On the left-hand side, the fast metabolizers, absolutely no increased risk, whereas the slow metabolizers, even 2 to 3 cups a day, is now associated with a significantly higher risk. Now, at the time, there was reason to believe that caffeine might act as a trigger for a heart attack, more so among younger cases. So when we looked at those below the median age of 50, you can see the y-axis will now change, and the slow metabolizers that drink 4 or more cups a day have almost a 4-fold higher risk of having a heart attack. On the left-hand side, with the fast metabolizers, you can now see a significant protective effect with moderate consumption. So because caffeine is the only major substance in coffee that's detoxified by CYP1A2, this strongly implicates caffeine as a trigger for a heart attack. Now, it wasn't until a couple of years later that another group in Italy essentially replicated these findings, but looking at hypertension. Now, the cup sizes are a little bit different and the categorization is slightly different, but nevertheless, you see the same pattern. On the left-hand side, fast metabolizers are protected. On the right-hand slide, slow metabolizers, you see a higher risk. Now, the recommendations for coffee consumption, certainly by Health Canada, you can consume up to 4 cups a day. Recently in the US, similar recommendations. This is just an article from the Washington Post that came out a few months ago after those guidelines and said, hey, these recommendations may in fact be wrong because you're overlooking a lot of this evidence that suggests, in some cases, half the population might be slow metabolizers, and you actually can't just be giving everyone recommendations to consume up to 4 cups a day. This is just a quote from the article in the Washington Post by an epidemiologist who noted that there are spectacular differences in how people metabolize caffeine, and to expect that everyone will respond the same way is absurd. So this just illustrates this whole concept of personalized nutrition as part of the broader umbrella of personalized medicine, where one size clearly does not fit all. The challenge, of course, is how do we balance the advice that we give to individuals with the population-based recommendations that we give to everyone. Well, there's been a proliferation of these consumer genetic testing companies that claim to provide personalized recommendations based on your genetics. Many of these are offered direct to consumer, but some are offered only through healthcare professionals. We thought that's an interesting question. We don't know what happens if we give people this kind of genetic information. On the one hand, someone might say, I have the risk gene, I'll be careful of what I eat. The flip side could be something like this. I have the gene, there's nothing I can do, leave me alone. Now, this scenario really applies to the concept of disease risk genes. When it comes to the modifier genes, there is something you can do. If you're a slow metabolizer and you drink lots of coffee, you can actually cut back. If you're told you have the gene for salt-sensitive hypertension, that might motivate you to limit your sodium consumption. It doesn't mean that the gene alone will predict risk of hypertension. It just means the way you metabolize sodium is such that you may actually have a higher risk of hypertension with higher sodium consumption. So we did this randomized controlled trial and found that giving people this kind of information does in fact motivate them to want to change their eating habits, and they actually understood the information to a greater extent. When we looked at the 1-year follow-up, we found that those who received the DNA-based dietary advice did in fact change their eating habits to a greater extent. So to summarize then, is DNA-based dietary advice ready for prime time? I think the answer now is yes. We have many examples of these gene-diet interactions being replicated. I've only illustrated this with the one with caffeine and CYP1A2. I haven't shown you examples of how ethnic background doesn't really matter if you're looking at the right genetic marker. Certain genetic markers are much more common in some ethnic groups than others, but at the end of the day, it really comes down to the individual. We've shown now that using the gold standard RCT design, that you can actually motivate people to comply with the dietary advice that you give them, and more importantly, it's the people that need to listen to this advice the most. Of course, the information has to be actionable. You're not just telling someone you have the increased risk of a disease based on their genetics and then not giving them any kind of advice. So that's really where there are opportunities in this field. And there's also greater consumer awareness and demand for this kind of information, along with this paradigm shift away from disease treatment and focusing more on wellness and prevention. So these are just some of the sources that have funded some of our research at U of T. And I'll just leave you with this quote. Even though the term nutrigenomics is fairly new, the concept is quite ancient. And Hippocrates noted over 2,500 years ago that positive health requires a knowledge of man's primary constitution, which is really just another way of saying genotype. And he goes on to say the powers of various foods, both those natural to them and those resulting from human skill. And clearly here he was referring to the concept of personalized nutrition. Thank you for listening.